So now that we're in week 10, we're finishing up this quarter here. I'd love some feedback from you in terms of, of all the subjects, what was your most favorite topic or set of topics during the quarter? So as you know, the quarter was divided into 3/3. Let's see. How about loved all equally? Here we are. All right. So the first third of the course, we covered evolutionary mechanisms and speciation and so on. During the second third of the course, we walked through some of the taxonomic groups. Now we're in the first third, second third, third third. Now we're covering ecological concepts. Go ahead and click in. Maybe we can talk about this in more detail in the office hours as well. Okay, has everybody clicked in? Alrighty. Let's see what you guys think here. The Bio94 instructors get together and chat about what topics we cover and why and so on. That's interesting. So the first two were the favorite, particularly the first one. Fewer ecologists in this group. All right. That's interesting. Thank you. That's helpful feedback. So fair enough. Okay, you picked your favorite. Here we go. So this is the same question, except now instead of picking your favorite, please pick your least favorite. So I guess all three are tied for least favorite. Let me know. Go ahead and click in, and I'll show you what your peers think. Okay, has everybody had a chance to click in? Let's see, which was your least favorite? Let's see here. Taxonomic group. Interesting. It's interesting. Clear differences between the 1 p.m. and 3 p.m. class. That's very interesting. Well, it's your feedback, and again, obviously it'd be lovely to continue this conversation in more detail as well, but so please feel free to speak up during office hours. We'll go ahead and get started then on the next topic of community ecology, since I have no way of knowing if you've actually been able to finish the population ecology or not. This information will not require that you've actually listened to the population ecology section of the course or not. So what is community ecology then? This is defined as the study of how species interact within communities. So then we have to consider what's the definition of a biological community, and a biological community can be identified as the populations of various species that are all living together with potential interaction in the same area. So, let's start by considering how these species interact then. And we're going to start by considering an example actually that's straight out of the population ecology chapter. I'm choosing to discuss it now within the context of community ecology rather than in population ecology, because I think it actually makes a fantastic transition between the subjects of population ecology, where we're considering one species at a time, and making the transition to community ecology, where we're really focusing on the interactions between different species. So this is figure 54 and 9 from your textbook in front of you right now, and shows the way in which the populations of two different species that live together undergo these rather extraordinary cycles. The cycles are shown here in this graph. The x-axis is time in years, and we have the population sizes for two different species plotted on the same graph. So the two species are snowshoe hares. The y-axis is shown over here on the left, and the line that corresponds to the hare populations is shown in gray. And the lynx is a member of the cat family, and it's shown as a blue line as shown here on the same graph. Now the relationship between lynx and snowshoe hares is shown here in the image at the top of the screen. The lynx are carnivores, they're predators that actually specialize in snowshoe hares. So these are the prey items, this is the predator. Snowshoe hares are herbivores, they eat vegetation. So, ecologists who've been studying the populations of hares and lynx, and where do these live, by the way, they live in northern Canada, and specifically in the biome of the boreal forest, which is now a familiar biome to you. Okay? Ecologists who were studying these species noticed that that the populations underwent these huge changes in how numerous the individuals of these species were in the area. So if we look at the hares first, for example, this gray line here, and up and down, and up and down, and up and down, and up and down, and so on, cyclically like this, and similarly the lynx, up and down, and up and down, and up and down, and up and down. Now, in natural populations, some populations, depending on what species, so not considering these two species, but species in general, some are relatively constant, some fluctuate somewhat unpredictably. Others do, in fact, show cycles. This is a really classic example of a very strong cycle that is seen in two different species that interact. So you can see a couple of different things from this graph. One is that both of them cycle on a sort of comparable time scale, namely on the order of about 10 years, meaning that the great increase and then crash occurs again repetitively at about 10-year intervals or so. That's true for both the hare population and the lynx population. And you should note, by the way, that the scale is completely different for the hares versus the lynx, right? The top number over here in the lynx is 25 per 100 square kilometers, and over here it's 4,000 per square kilometer. So there's far more hares per unit area than there are lynx. The lynx populations also cycle approximately at 10-year intervals, but they're phase shifted relative to each other, meaning they don't hit their peak at exactly the same time. And specifically, the hare populations tend to peak about 2 years before the lynx population peaks. The hare population crashes, and then about 2 years later, the lynx population crashes as well, meaning goes to much smaller numbers and then rebounds again. So ecologists, of course, were very interested in understanding what is driving these cycles. So one can generate a number of hypotheses from comparing what's going on with their natural history. Of course, if hares are the food for the predator, then one could assume, for example, that perhaps what's happening here is that the hares, as their population gets very large, maybe they eat all the vegetation in the surrounding areas, and then there's no longer enough food to sustain the population, so it undergoes this huge crash, and now there's not enough food for the predator, the lynx, and so they crash in response to what's going on with the hares. That particular scenario would then suggest that it's driven primarily by the food availability to the hares is driving both of these cycles. Another possibility would be that as the hare numbers grow up, then this is able to support a larger and larger number of predators, but after the predators get very numerous, then perhaps their increasing numbers cause the hare population to crash. And once the hare population crashes because of the predation pressure by the lynx, then the lynx start to have a population crash in addition because there's now less food for the population of lynxes. So what's driving what, and how can you figure this out? Well, this has actually been experimentally addressed, and the logistics of such experiments are quite tricky, right? You're talking about phenomena that occur over fairly long cycles, 10-year cycles, and so that means you have to be out there for years to collect data to try to understand this. You're also trying to follow populations that are constantly changing in terms of their size. So how was this done experimentally? Well, the ecologists who tackled this problem set up a number of experimental plots in the boreal forest, northern Canada, and some of the plots were controls, namely they were not manipulated in any way, and they could just go visit these controls and monitor the background hare and lynx populations. Then in the experimental treatments shown over here on the right, some of the plots had electrified fences put up that would keep the lynx out, and so the hares in this area then would be relieved of predation pressure. And so if one saw a change in the populations in this plot, then this would be in response to a decrease in the predation specifically. There was other plots where the manipulation was to provide extra food, that's specifically for the hares, extra food for the hares. but no electrified fence, so the lynx were still able to come in and prey on the hares. And then finally there were plots where the lynx were kept out and there was extra food for the hares. The ecologists went in year after year after year and did sampling methods that are now more familiar to you, such as mark and recapture, and other methods to evaluate the number of hares. And this is what they found. So the results are plotted as the number of hares in the experimental plots relative to the number of hares in the control plots. That is, it's a ratio where they divided one number by the other number. So the reason that they did this, again, was because in the control plots, the numbers are going up or down depending on where they are in the cycle, right? So it's very difficult to compare numbers with each other if they're constantly going up or down, which is why they were always compared as a ratio relative to the control plot, right? Does that make sense? So a ratio of 1 then would mean that they were exactly the same in the experimental plot as in the control plot. That's what a ratio of 1 would mean. So the 3 different kinds of treatments are shown here in 3 different graphs, and on the x-axis here is time in years. Anything above 1 means there's more hares in the experimental treatment. Anything below 1 is more hares in the control. So you can see, eyeballing this, that the results are pretty consistent, that in all of the treatments, some of the numbers are quite close to 1, but if there's a deviation from 1, then it's always in the same direction, meaning more hares in the experimental treatment. Let's start over here on the left to consider the case where there are no lynx. So the food available is still the same, but they're just no longer being preyed upon by the lynx. You can see that in this particular case, there is in fact an increase in the number of hares in these plots relative to the control. So that suggests that yes, there is a response to predation. Going on to the next graph here, extra food. When there was extra food, this also resulted in an increase in the number of hares. So that suggested that yes, they were sensitive to food availability as well in terms of the population dynamics of the hares. And then finally here in the last plot treatment, you see the effects of both at the same time. And it may strike you that this effect of this particular combination of treatments is much stronger than the sum of the other 2 effects, right? So if you add the effects of no lynx and the effects of extra food and add those up, you get a bar that would be much smaller than the actual height of these bars. So these 2 working in combination actually have a much, much stronger influence on the hare numbers. And so the ecologists concluded then that it's inappropriate to look at this as either being driven primarily by top-down pressure from predators or bottom-up food supply to the hares, but rather both of these factors were really important, and that these cycles are being driven primarily by interaction between these 2 different species, both the predators and the prey. So community ecology, then one is primarily concerned with interactions between different species, such as the predator-prey example that we just walked through. There are different kinds of interactions that are possible between different species, and they're typically characterized by the overall effects on the individual 2 species that make up the association. So the interactions are shown here within parentheses. This is the typical way. So plus minus, for example, would mean that one species had a positive effect and the other one had no effect. And what is it exactly that's being scored? Survival or reproduction or fitness? Because ultimately, of course, fitness is the relevant currency here. And so survival or reproduction of the 2 interacting species is what's being compared here. So commensalism, competition, consumption, or mutualism are 4 of the main different kinds of ways in which species can interact. Commensalism, starting at the top here, is a name given for an interaction between 2 species, in which one species benefits and the other one there's no perceptible effect whatsoever. I'm not going to say very much about this. There's a very short section in your textbook. I'll go ahead and let you just read about that. I'll spend much more time on competition. Now, there's much more known about competition. So let's go ahead and consider competition then as an example of an extremely important interspecific interaction. So competition is coded as minus, which means that you can identify competition between two species if the presence of either species will have a negative impact in terms of survival or reproduction, some measure of fitness on the other species, and it's mutual in terms of each one is negative. One way of describing this, or identifying this, is to say that, well, these species are competing for some kind of limited resource. So it's not just that there is some resource that they're both using, but it has to be limited, or one species will not actually influence, it will have any effect on the other species as a whole. So what are some examples of limited resources? Things like specific food types, space, or even access to sunlight for photosynthetic organisms. These are examples of limited resources. Ecologists have described a couple of phenomena that result from competition. One of the phenomena is called competitive exclusion. And this is a phenomenon that's given this name if, when two species are competing for limited resources, that this competition actually leads to local elimination of one competing species, in which case that species that's eliminated would be considered the weaker competitor. This is also sometimes described as the so-called competitive exclusion principle, which is just simply a statement that two species that are competing for the same limiting resource can't coexist permanently. If they do exist in the same place, this is usually temporary. And so this is concluded because it's extremely unlikely that in the real world that two species are equally good at competing for resources. And so whichever species has some kind of tiny advantage over time will be able to move ahead of the other species. This recognition that coexistence between two species that are going after exactly the same limiting resource then led to articulation of a concept that's called the niche, or the ecological niche. So what a niche is a description of the resources that is used by a particular species. So when a species niche is being described, then this is the various resources that that species uses. Putting these two things together, the competitive exclusion principle and also the concept of the ecological niche, if we sort of put these two thoughts together then, one can summarize this as a statement that two species can't coexist if they occupy the same ecological niche. So let's examine what this would look like in a graphical representation. This is figure 55-2 from your textbook in front of you now, and is an example of a potential limiting resource to have something concrete to think about when one is thinking about a niche. The x-axis here is seed size, going from smaller seeds on the left and larger seeds over here on the right. And so seed size is an example of something that could be considered a niche because many organisms specialize on seed sizes. And so the representative individual here, or species here, so if this curve here represents the consumption of members of species 1, then you can see that members of this species on average eat seeds of about this size, but eat seeds as small as this size and as large as over here. So there's some range of seed sizes that this species will actually eat. So now let's bring in a second species and see what happens. Okay? If now there's a second species, go ahead and put it right here. The second species eats seeds in a range that overlaps completely with species 1. That is, the complete range for species 2 is within the range of species 1. So what's going to happen now? Well, it depends which one is the better competitor. If, in fact, species 2 is a weaker competitor for the seeds, then it should make sense to you that species 2 will be eliminated locally, right? It'll be outcompeted. It'll be extremely unlikely that species 2 can actually coexist in the same area with species 1. Does that make sense? So the expectation would be then that it would be gone. Okay, let's consider something slightly different now. Instead of having a complete overlap, let's now assume that species 2 looks a little bit more like this, where there is some overlap, right? So here's the area of overlap in some sort of limited resource, but there's not complete overlap. What would happen in these conditions? Well, there's a couple of different things that will happen, and it depends if we're considering this more sort of short-term or long-term. So I'm now going to jump to a new diagram here for a discussion of those concepts. So I'm going to go ahead and use the same visual backdrop to sort of summarize the information that's in 3 different figures from your textbook to kind of try to draw these concepts together here, because they all have to deal with niche overlap, competition, and what happens when you have species with overlapping niches that are competing for limited resources. Okay, so let's again consider this blue line as indicating the niche occupied by species 1. And notice that the x and y axes have changed a little bit. We've now gone from seed size, which was a example of a limiting resource that could be part of a niche, to a more general, just conceptual niche. If it helps you to think about this in terms of something like seed size or area of space or something, then you can mentally substitute that in. This can be any kind of limited resource within the environment for the species. And the y-axis is now numbers of individuals using those resources. So let's bring in our second species here. Here's species 2. Well, let's assume that just as in the previous example, that species 1 is the better competitor. Then what would we expect from this particular distribution of occupation of the niche space by these 2 different species? Well, it looks like species 1 will be able to use the resources in the area of niche overlap, and species 2, and I'd be relegated to the space that is not being used by species number 1. Does that make sense? This relates then to the definitions of the differences between a niche that could be realized in the absence of the interactions with the other species, and the small subset of a niche that can be occupied because of interference with other species. So this is the difference between fundamental and realized niche. So the fundamental is the hypothetical niche in the absence of the other species. So for species 2, this would be the solid line then. The fundamental niche would be this region right here, fundamental niche for species number 2, right? Would be that whole range. That would be the range of resources that could be used by species number 2 if it wasn't being interfered with by species number 1. The realized niche, if we use that as the dotted line, then that's this area right here. Should make that dotted. I'm going to erase that. This would be the realized niche right here. Does that make sense? The realized niche always has to be smaller than the so-called fundamental niche because it's the part of the hypothetical all-inclusive area that can be used by the species in the absence of interference by the species. And the realized niche can just be a subset of that. So that's the difference between realized versus fundamental niches. Over longer periods of time, go ahead and get a new longer periods of time, meaning over many generations, one can expect evolutionary changes in the populations that are in contact here that will lead to differentiation in the niches such that over a long period of time, then you could imagine that evolutionary changes would cause species 2 to have a very different niche in the area that is occupied in proximity or overlapping than with species number 1. Going through a concrete example might help make that concept a little clearer. So let's go ahead now and consider some experimental evidence or manipulations that help scientists evaluate fundamental versus realized niches and when niche differentiation is actually occurring. So we'll start by a classic example of a manipulation in the intertidal zone. This is figure 55-5 from your textbook in front of you right now, and shows a diagrammatic representation of some of the kinds of organisms that one finds in the intertidal zone. So this is the area where seawater meets land. Specifically, this is the rocky intertidal, and what we have here are a couple of different species of barnacles. Barnacles are within phylum Arthropoda, right? So this is the phylum that includes the crustaceans and the insects and the millipedes and spiders and scorpions and all sorts of other creatures. Barnacles are highly unusual arthropods in that they are sessile as adults. In fact, they are even attached to the substrate as adults. So as immatures, they're planktonic, and then they land on a hard surface, such as the rocks shown here. They settle, they attach themselves to the rocks, and then they live there for the rest of their lives. They grow bigger and bigger, but they don't move around on the rocks. They remain attached in place, and they are, in fact, filter feeders. That's how they make their living. Very unusual arthropods. Most arthropods are not sessile attached organisms. Anyway, in the intertidal zone, one often gets bands of different kinds of organisms up and down, going farther away from the water and farther down into the water relative to the mean tide level. So in this particular diagram, then you can see the mean tide level is indicated by this line right here. So as the tide goes in and out, then the seawater level goes up and down, and the farther down into the water an organism is, the more time that organism spends submerged in seawater, and the higher an organism is in the intertidal, the larger fraction of time that organism spends exposed to air. So within the intertidal, then, one does get these bands of different organisms at different heights, as shown here, also for the barnacles. So there's 2 genera of barnacles that are listed here. Thamellus, which is primarily found in the upper intertidal, is shown here, and Semibalanus, which is another barnacle genus. And these species are found typically in the lower intertidal, as is shown. So the ecologists are very interested in, well, are these different species hanging out in different parts of the intertidal because they have very different requirements in terms of some factor in the environment? Or are they actually in competition, and one is keeping the other one from expanding into the area? How can you tell? Well, the ecologists did some experiments which are diagrammed right here. Because these organisms are attached, that makes it a little trickier to move them around. So what they did was they took rocks from the upper intertidal that already had Thamellus individuals on them. They physically moved them and bolted them into the lower intertidal and allowed some Semibalanus planktonic larvae to settle on the rocks and become attached and start growing. And then in these rocks, they divided them down the middle and removed the semibalanus from just one half of each of the experimental rocks, which meant that on these rocks then, on one half just the thamellus was left, and on the other half the rocks, both of the different kinds of species, which are also in different genera, were still on the rocks. And then they left them in the lower intertidal and looked to see what happened, right? So if thamellus just doesn't live well in the lower intertidal, then you would expect them to die off on either side of the rock, and it wouldn't really matter if semibalanus was there or not. And yet if semibalanus had some sort of effect, then you would expect to see an effect. So they went back later and found that in fact survival of these upper intertidal barnacles was severely influenced by the presence of a competitor. So that's shown as a bar graph over here. And when the competitor was present, when the semibalanus was present, then only a very tiny fraction of the thamellus actually survived. If they were absent, then they did fine. So there's nothing about the lower intertidal per se that is keeping the thamellus out of the lower intertidal. It is specifically the actions of the semibalanus. You might think, well, if they're just attached to the rocks, how could these semibalanus possibly impact these likewise attached thamellus? They apparently were able to impact them as they grew in place. They are attached, but as they grew in place, they were able to sort of grow into them and then dislodge them off of the rock. So that's how they were able to kill them. So that's the influence of thamellus, on semibalanus on thamellus. But how about the other way around? Right? This was just a one-way experiment where rocks were taken from the upper intertidal and brought down here into the lower intertidal. I was mulling over this and thought, well, this would actually make a good clicker question. So here we go. I'll go ahead and activate this as a clicker question. So here it is. I tried to squeeze the clicker question into this otherwise crowded. There we go. So here's the clicker question. Oh, is that people's phones?
Yeah, it's an Amber Alert, and they send it out on everyone's phone.
Oh, I see. So that's the... Yeah. I don't have a cell phone on me. I left my cell phone in my office, so it's not me. Thank you for explaining that. All right. So, the question is, can you tell the boundaries of the fundamental niche of semibalanus from these experiments, right? So that's different from asking about thamellus, right? And so before we were distracted by the alarm, we can consider, well, thamellus exists here, right? So this would be the realized niche right here. But given that the thamellus, in fact, could occur just fine in the lower intertidal, then we would consider the fundamental niche actually extended down into the lower intertidal, but that's for thamellus, right? How about semibalanus? From these experiments, can we tell where its fundamental niche is? Yes or no? All right, what do you think? Okay, you guys have all clicked in? All right, let's see what you think. Okay, looks like everybody's clicked in. Next you say, simply because one species has a particular kind of effect on the other doesn't necessarily mean you can infer that the reverse is true. Really, in order to evaluate what the fundamental niche is of semibalanus, you would actually have to do the same experiments in reverse. Take a rock with semibalanus, move them up into the upper intertidal, and so on. You really can't make inferences about one particular species from experiments that are done in another one. So, alrighty. So this is a nice example of what one would expect after some sort of long-term association between two species with overlapping niches. So this is figure 55.7 from your book, and shows the distribution of beak depths for two different species of bird. The bird head is shown here in the upper right, and this is for two different islands. I can't even tell where that's coming from. Can you guys tell where it's coming from? This.
And everyone's phone gets it.
Okay, so you guys can't turn your phones off, or whoever's phone is on that's doing this is. Oh, is it coming through the speakers? Good grief. Well, there's no phone up here. Sorry, guys. Well, I'll just forge ahead here. Okay. Back to the bird. Okay, the x-axis here is beak depth. Why do we care about beak depth? That's this length right here, right? You've actually seen a number of graphs throughout the quarter where beak depth is something that is measured and then plotted. And so by now, hopefully you're used to the idea that beak depth is a functionally significant morphological trait or attribute of birds because it has a huge influence on the kinds of food that that bird can actually eat. It relates for the kind of seeds that can be manipulated or cracked open or so on. So, if we start down here in the lower graph here, the y-axis is the percentage of finches that were sampled. So you can see with these two different species within the same genus, then there is a distribution such that there is basically no overlap. And so in the distribution of beak depths. And so because beak depth is sort of a proxy for the kind of food being eaten, then it would be logical to infer that these two species then are going after different food. And so the niches, at least in terms of food as a potentially limited resource, then there's basically no overlap between these two species. If one considers, though, what the distribution of the same trait is in this one of those species in the absence of the other species, these data actually exist because these two bird species don't always exist in close proximity. So on a completely different island, which is shown up here at the top, you can see that in the absence of this other species, which is shown in dark blue, the character distribution is really quite different. Being more intermediate between these two when they coexist. So such distribution patterns are often used by ecologists or evolutionary ecologists as a indirect evidence of past competition. Why past competition? Well, it's not present competition because right now there's virtually no overlap in the niches, at least as indicated by beak depth distribution patterns in these two species. Although, if one thinks about this as being the possible beak distribution pattern that would exist in the absence of competition from this other species, then there would certainly be a lot of niche overlap. So this phenomenon is referred to as character displacement because of the change or displacement in the magnitude of the trait. So for example, down here in this island, the character is more at 5 point something, and down here it's 4 point something. So this has been a displacement, a change in the actual magnitude of the trait of that particular character in response to competition. Okay, I won't say anything else more about competition at this point. So, consumption and mutualism. Those are the two last remaining examples of interspecific interactions. Consumption is pretty familiar to us as consumers. Within consumption, then there's a plus-minus interaction. This is the consumer is deriving a benefit from consuming another organism. So this is the consumed organism here, has some detriment to being consumed, right? So I'm not going to spend a lot of time going through some of the examples of this. I'm going to jump to, actually, yeah. I'm just going to go ahead and jump to an example of an interesting phenomenon that one gets that is driven by consumption patterns or interactions that are driven by consumption, and that is the phenomenon of mimicry. This is a term that is used when two completely different species have some kind of very strong resemblance to each other. One mimics the other. And so this is not used as a term to describe similarities that are just based on close relationships, right? So if two species are closely related, then you might think, well, of course they look alike because after all, they're closely related. Mimicry is a completely different phenomenon. This is an example of convergent evolution, meaning that the similarities are not driven by shared ancestry, but are driven by independent evolution of the same traits, whatever they are. Now, ecologists who are interested in studying organisms that mimic each other in the wild, and an example is shown in these two insects, which are not closely related to each other, and yet they're a very striking resemblance to each other. There are different phenomena that are different kinds of mimicry patterns, and so they're given different names. Let's go ahead and start with a mimicry phenomenon that is referred to as Batesian mimicry. In Batesian mimicry, one has a palatable species. Palatable means good to eat, is mimicking an unpalatable one, where the unpalatable one is maybe poisonous or tastes nasty, so distasteful, tastes horrible, or is full of poison, something. So if you have a species that is actually not poisonous and not distasteful, but it actually resembles an unpalatable one, then it will be afforded some level of protection from predators because they will avoid it, thinking that it's the poisonous or the distasteful one. And so this is a form of natural selection that can drive mimicry of a perfectly innocuous species for a poisonous one. Another example of what could be considered a Batesian mimicry would be some kind of non-dangerous species is mimicking a dangerous one and thereby having some level of protection as well. Now, of course, the level of mimicry has to relate to whatever kind of sensory system is being used by the predator. And most of the examples of mimicry are, in fact, visual. This is probably no accident, that's because we as humans are highly visual creatures, and so most of the examples in literature are, in fact, examples of visual mimicry, but in fact, mimicry can occur in any kind of sensory modality. And so it could be smell or what it sounds like, anything else. The other common kind of mimicry is called Mullerian mimicry, and this is really quite different. This is what happens when there are different unpalatable species and they evolve to resemble each other more closely. So this is more like reinforcing a message for a potential predator. So this is obviously different evolutionary steps would be required to evolve such a Mullerian mimicry system than a Batesian mimicry system. So you might want to think about under what conditions would you have evolution of these, and specifically in Batesian mimicry, what makes this work? You know, presumably it also depends on how many fakes are out there, how many mimics are out there that are actually perfectly good to eat. You know, there's only so many individuals that can be good fakes out there before the predators figure that out. So anyway, this is where I left off at 1 p.m., so I'm going to go ahead and stop right there. Perhaps I'll see some of you in office hours tomorrow, otherwise I'll see you Wednesday.